Hello everyone, my name is Gary O'Mealy, and in this video we are going to discuss osmosis. The transport of water across membranes, or osmosis, is just a applied kind of facilitated diffusion. So you're going to take everything that we talked about with facilitated diffusion in the last video, and you're talking about that just in the context of water moving across the membrane, right? So the idea here is that the reason why water has to move by facilitated diffusion rather than simple diffusion, yes, water is a very small molecule at 18 grams per mole, but as we've seen in the past, it is very, very, very polar, so it's not going to move across membranes by simple diffusion very well. So it's going to have to do it by facilitated diffusion. So it requires specific water channels, which are called aquaporins, and that is going to be what allows water to rapidly move across membranes. You can see this illustrated here. An aquaporin that is inserted into the plasma membrane of a particular cell will allow osmosis to occur at a high rate. Many cells in the body do possess these aquaporin channels, but in the future, especially when we talk about kidney function, you'll need to be aware that not every cell in the body has aquaporin channels, and those that do not have aquaporins are impermeable to water. So before we really get into osmosis, we need to consider a few things. Number one, water is just like any other molecule in that when it diffuses across membranes, it's going to want to do this from areas of high water concentration to areas of low water concentration. Additionally, because water is the solvent and not one of many particular solutes, this is a little bit more complicated because very rarely are you going to be directly told what the water concentration is. In the case of the solutes that we talked about in the previous video on passive transport, determining which direction that molecule would want to move based on the concentration gradient was easy to determine because we were directly told what those relative concentrations were on either side of the membrane. What we're saying here is that because water is the solvent, we're not necessarily going to be directly told which side has more water or which side has less water. We are going to need to infer that information from some important clues. So look at this example here. We have two different containers that both contain a simple solution of sodium chloride. However, the left container contains a much lower concentration of sodium chloride at 1%, and the right container contains the same solution, just 10 times as concentrated. You can see each container contains an equal volume of solution, but the question we want to ask here is, in which solution is there a higher concentration of water? If you look at the left solution, 1% sodium chloride, if sodium and chloride ions are literally the only solutes that you find in this solution, that means that the remaining 99% of the solution has to be made of solvent, in that case, water. Whereas the other solution, which was 10% sodium chloride, the other 90% has to be water. So clearly in this case, the leftmost container has a higher concentration of water compared to the rightmost container. Our conclusion from this little thought experiment is that higher solute concentrations are going to mean lower water concentrations. So because we are not going to be directly told what the water concentration is, we need to infer the water concentration from looking at solute concentration. Some more osmosis considerations. Water, as we said, will want to move from areas of high water concentration to areas of low water concentration. To make this more practically useful for us, though, since we are going to be told what solute concentrations are, water will move from areas of low solute concentration to areas of high solute concentration. That is exactly the same thing as saying water will move from areas of high water concentration to areas of low water concentration because of what we established on the previous slide. Next, water will only move if the water imbalance, the concentration gradient for water we mean, cannot be corrected by the movement of the solute. So to really understand what point number 4 here is saying, we need to do another little thought experiment here. So look at this setup that we have here. We have one giant container that is split into 2 different chambers by the presence of a vertically oriented semi-permeable membrane. The solute that we have in the 2 chambers we can say is glucose, and we'll go ahead and say that glucose is the only solute that is present here. And again, that semi-permeable membrane splits the entire container up into chamber A and chamber B, as we will define them here. Now, clearly, we can see that chamber A has a low concentration of glucose compared to chamber B. As it currently stands, there are concentration gradients across the membrane for both glucose and water. There is a high concentration of glucose in chamber B, so what glucose would really like to do is diffuse across the membrane from chamber B into chamber A until we establish equilibrium. So the idea here is that if the membrane is permeable to glucose in this case, then both the glucose and the water gradients can be addressed just by diffusion of the solute. So if we allow the glucose to move across the membrane here until we reach equilibrium, now at equilibrium, there is no concentration gradient for glucose, and there is no concentration gradient for water. There would only be a concentration gradient for water if there is a solute concentration difference across the membrane, and clearly there is not. So this is an example of a case in which osmosis does not need to occur because we can address the water gradient by moving the solute instead. But in a lot of cases, we're going to need to consider possibilities of, well, what if the membrane is not permeable to the solute? What if this glucose cannot move across that yellow membrane? So the inability for us to relieve both the glucose and the water gradients here, this is going to cause a buildup of osmotic pressure. All that extra water that's on the left side of the membrane is going to push against the membrane, causing, if this were a real cell, it would cause the cell to kind of swell up a little bit. So after we allow water to move across the membrane, because glucose cannot, you'll notice that the volume of solution in each chamber has changed. The volume in chamber B has gone way up, and the volume in chamber A has gone way down. But you will notice that the number of glucose molecules in each chamber is exactly the same because, as we said, in this particular example, glucose cannot move across the membrane, but water can. So the idea here is that as long as the membrane is permeable to water, in practical situations that's going to mean that the membrane has aquaporin channels, water will move across the membrane until equilibrium for both water and solute is reached. Now, the thing you have to remember here, because probably what you're fixated on right now, is the fact that chamber A and chamber B still contain very different numbers of glucose molecules. But gradients and transport are not based on the total number of particles. It's based on the concentration. Concentration is equal to the number of particles per unit volume of solution. So even though not a single glucose molecule has moved and chamber A has a lot fewer glucose molecules, both chambers now have an equal concentration of glucose because of the reduction or increase in volume in each chamber. A reduction in volume will increase concentration, and an increase in volume will reduce concentration. So the number of particles in each chamber did not change, but both concentrations did change because they became equal because of the change in volume. That is establishing equilibrium for both solute and water. So you'll notice that each glucose molecule is about equally spaced apart, which is as good a measure of any of concentration. Highly concentrated molecules will be very closely packed together. Very sparsely concentrated molecules will be very far apart. So now that we've got that out of the way, let's finish up our conversation on osmosis. Water concentration is determined by total solute concentration rather than the concentration of any individual solute. Now, this is a very important point for us to make because the previous two examples we've looked at are situations involving solutions that only contain one solute. Whether that solute is glucose or sodium chloride, we were only really looking at one solute. But the thing you have to remember is that in the body, our fluids, our body fluids, blood plasma, intracellular fluid, etc., these are complex solutions that contain dozens and dozens of different solutes that are all going to affect what the total water concentration of that fluid is. So the term that we're going to introduce here that refers to the total solute concentration in a fluid is called osmolarity. So to illustrate how to calculate the osmolarity of a bodily fluid, let's look at this solution here. This container contains 5 different types of solutes in a water-based solution. The concentration of each individual solute can be expressed in terms of molarity, which is the number of moles of that solute per liter of solution. The water concentration, as we've said, is determined by the total concentration or osmolarity of these 5 solutes put together. So if we're going to determine this solution's ability or capacity for undergoing osmosis, that's going to be something that we're going to need to calculate. So let's say, for the sake of argument, the concentration of solute A is 10 millimolar, the concentration of solute B is 20 millimolar, for C it's 5 millimolar, for D it's 10 millimolar, and for E it's 2 millimolar. If we're going to calculate the total osmolarity of this solution that contains 5 different solutes, we're just going to add those individual molar concentrations together. In which case, if we do that, we get an osmolarity value of 47 milliosmolar. Now, it's important to note that this number does not really mean a whole lot to us in terms of osmosis just by itself, but what this number is going to allow us to do is quickly infer the relative water concentration in this solution relative to another solution that is presumably on the other side of a membrane. In determining whether osmosis occurs or not and in which direction osmosis occurs, you're always going to be comparing the osmolarities of the fluids that are on opposite sides of the membrane. Let's get some practice with that. So in chamber A here, you have a solution with an osmolarity of 100 milliosmolar, and in chamber B, you have a solution with an osmolarity of 200 milliosmolar. The idea here, as we've established before, is that solutions with low solute concentration or low osmolarity, they have higher relative water concentration, and vice versa. So what this is going to mean is that because there is a water concentration gradient here, and since presumably none of the involved solutes are going to move, water will diffuse from the area of low osmolarity to the area of high osmolarity. So from left to right, chamber B would increase in volume, and chamber A would decrease in volume. Look at this situation instead. The osmolarity of chamber A and the osmolarity of chamber B are equal to each other. They are both 200 milliosmolar. If the osmolarities on both sides of the membrane are equal, then no net osmosis is going to occur. And the word net there is particularly important because this does not mean that water will not move across the membrane at all. It just means that the rate of osmosis from right to left is equal to the rate of diffusion from left to right. So they basically cancel each other out. So there will be no net change in volume in either chamber. One last thing for us to consider about osmolarity is that when you're calculating this, you are only going to count the so-called non-penetrating solutes. Those are the kinds of solutes that cannot move through the membrane by simple diffusion. So we're going to be talking about things like ions like sodium and potassium, sugars like glucose and fructose, amino acids, things of that nature, either because they are too polar or because they are electrically charged. One of the major rules that we discussed about osmosis is that water will not move if the solute is capable of moving all by itself. So, if we're calculating osmolarity, that being the case, it wouldn't really make sense for us to count the so-called penetrating solutes like oxygen gas, carbon dioxide gas, things that can move through the membrane in any situation anyway. So when you're calculating osmolarity, you will exclude from your calculations any solutes that can freely move through membranes by simple diffusion. Again, we're talking about things like oxygen, carbon dioxide, lipids, steroid hormones. Those concentrations in solution we're not really going to worry about. And then finally, a very important number that I would like you to commit to memory. I'm not going to ask you to memorize too many things, but when I do ask you to memorize something, you'll know that it's a very important thing for you to remember. The standard osmolarity of most human bodily fluids, including the blood plasma, is 300 milliosmolar. This is a very important number for you to remember because this will always serve as a reference point for you to determine whether osmosis will occur in one scenario or another. If you are looking at a situation in which the osmolarity of the blood plasma is different from 300, then you'll know that osmosis is going to occur in that case. Okay, that's going to do it for this video. Here is a list of vocabulary terms you probably ought to be aware of. And for checking your understanding, number 1, what purpose do aquaporin channels serve? Number 2, how can we infer water concentration from total solute concentration? Number 3, under what circumstances will osmosis occur? And finally, what is the set point osmolarity for most human bodily fluids? And it's important to understand that this is regulated by homeostasis, and we will talk a little bit about that in the next video and in the future. All right, that's going to do it for this video. If you have any questions, go ahead and leave them in the comments section, and I will see you next time. So long.